haunted and fascinated humanity for millennia. Can we live forever? The desire to cheat death to extend our precious time on this earth is woven into the very fabric of our history. From the epic tales of ancient heroes searching for mythical fountains of youth to the whispered secrets of alchemists promising elixirs of life, the dream of immortality is as old as civilization itself. This isn't just a fanciful notion from dusty old books. It's a deep, primal urge. Beauty loves discovery, and we naturally want to experience as much of it as possible. The thought that this vibrant existence has a definitive end date is, for many, a difficult truth to accept. This ancient yearning wasn't just about avoiding the inevitable, preserving wisdom, continuing a legacy, holding on to the relationships we cherish. Imagine the stories a person who had lived for centuries could tell. Think of the knowledge they could accumulate and pass down. Gilgamesh and the Sumerians' search for an eternal plant. Long before the advent of modern medicine, the pursuit of longevity was the domain of the alchemist. These mysterious figures, often working in secret, they were the chemists, metallurgists, philosophers of their time. Their ultimate goal was a grand one: the discovery of the philosopher's stone, turning base metals into gold, and, more importantly, creating an elixir of life. This elixir was believed to cure all diseases, rejuvenate the old, and grant the drinker a state of eternal youth. It was the ultimate prize, a solution to human mortality, a goal that consumed countless lives. The search for this elixir took many forms across different cultures. In China, Taoist alchemists, cinnabar, or mercury sulfide, held the key to immortality. They spent centuries developing complex processes to purify and ingest these substances. The turn of the 20th century marked a profound shift in our quest for longevity. The age of elixirs began to fade. Mythical fountains began to fade, replaced by a new era of scientific inquiry. The focus moved from the crucible of the alchemist to the microscope of the biologist. Instead of searching for magical substances, scientists began to look inwards, at the very building blocks of life itself. The germ theory of disease, established in the late 19th century, had already shown that invisible microbes could cause illness and death. This was a revolutionary idea. Our fate was not just in the hands of the gods, but was influenced by tiny organisms we couldn't even see. This opened the door to a new way of thinking about aging. One of the first major theories to emerge was the rate of living hypothesis, proposed in the early 1900s by Max Rubner, and later expanded by Raymond Pearl. They observed that larger animals with slower metabolisms, like tortoises, tended to live much longer than smaller animals with fast metabolisms, like mice. The idea was simple and intuitive. Perhaps we each have a finite amount of metabolic energy to burn through in our lifetime. Living fast, they suggested, meant dying young. This theory, while now considered overly simplistic, was a pivotal step. The second half of the 20th century brought a discovery: the unlocking of the structure of DNA in 1953. James Watson and Francis Crick revealed the double helix, the elegant blueprint for every living organism. This was the moment science fiction started to become scientific fact. If aging was written into our biology, perhaps the instructions could be found within our genes. The search for the genes for aging began a quest that still drives research today. It was a paradigm shift from external factors to our own internal code. In the 1970s, Elizabeth Blackburn and colleagues found tiny protective caps at the ends of our chromosomes. Picture them as the plastic tips on a shoelace. Every time a cell divides, telomeres shorten. After many divisions, they get so short the cell enters senescence, cellular old age. This gave a mechanical explanation for cellular aging. It suggested lifespan could depend on initial telomere length and the rate they shortened, a built-in cellular clock. Studies in simple organisms like yeast and roundworms revealed genes that influence lifespan. In the 1990s, Cynthia Kenyon's work on C. elegans was a watershed moment. A mutation in DAF2 could double the worm's lifespan. While geneticists were busy decoding our DNA, another group of scientists was making equally remarkable discoveries, armed only with weighing scales and measured portions. The idea that eating less could extend life has roots in historical traditions. But in the 1930s, Clive Mackay at Cornell provided the first hard evidence. He ran landmark experiments on rats fed severely calorie-restricted but nutrient-rich diets. And many lived significantly longer, some by as much as a third, than well-fed rats. For decades, the results were a scientific curiosity. The mechanism was a complete mystery. How could near starvation be good for you? Research picked up with studies across many species: yeast, spiders, fish, dogs. The pattern was consistent: cutting calories without malnutrition, extended lifespan. The most famous work involved rhesus monkeys. Long-term studies from the late 1980s showed CR extended their lifespan and delayed age-related diseases like cancer, diabetes, heart disease. So how does it work? Modern science is starting to answer that. Today, we stand on the cusp of a new revolution in longevity science, one that combines our knowledge of genetics, metabolism, and cellular biology, cellular biology, to develop interventions that were once the stuff of science fiction. Labs are moving beyond observing aging to actively reprogram it. One of the most exciting fields is senolytics. These drugs seek out and destroy senescent, zombie cells. Zombie cells stop dividing but refuse to die. They linger in tissues, release inflammatory signals that harm neighboring cells and drive age-related disease. Clear the clutter, rejuvenate tissues, restore youthful function. Early mouse studies are spectacular. Senolytics improved kidney function, restored fur, and extended lifespan in old mice. Human trials are now underway. While the cutting-edge science of senolytics and genetic reprogramming is incredibly exciting, the good news is you don't have to wait for a futuristic pill to start extending your healthy lifespan. Many of the most powerful tools for longevity are available to us right now, and they start with what we put on our plates. The evidence is overwhelming that diet is a cornerstone of a long and healthy life. One of the most consistently beneficial eating patterns is the Mediterranean diet. Fruits, vegetables, nuts, legumes, whole grains, healthy fats like olive oil, smaller amounts of fish and poultry. It's more of a lifestyle than a strict regimen. This way of eating works wonders because it tackles aging from multiple angles. The abundance of colorful plants provides a vast array of polyphenols and antioxidants, which help to combat chronic inflammation and oxidative stress. The high fiber content feeds a healthy gut microbiome, which we now know is crucial for our immune system. A healthy diet is a critical piece of the longevity puzzle, but it's only one piece. To build a truly robust foundation for a long and vibrant life, we must look beyond the kitchen. Perhaps the most potent anti-aging medicine we have is exercise. The benefits of regular physical activity are almost too numerous to list. It strengthens your heart, improves circulation, builds strong muscles and bones, enhances insulin sensitivity, and reduces the risk of chronic disease. Crucially, exercise is not just about high-intensity workouts. Cardiovascular exercise, brisk walking, cycling, swimming, resistance training to maintain muscle mass. Muscle is your metabolic sink. Maintaining it as you age is one of the most important things you can do for your health. Sarcopenia, or age-related muscle loss, is a major driver of frailty and disability. Resistance training combats this. Weights, resistance bands, body weight exercises. We have journeyed from the mystical dreams of ancient kings to the frontiers of modern genetic science. The quest to extend human life, once a matter of faith and folklore, is now a serious scientific endeavor, yielding discoveries that are reshaping our understanding of life and death. We are on the brink of having tools, senolytics, epigenetic reprogramming, targeted pharmaceuticals that could not only extend our lifespan, the number of years we live, but more importantly, our health span, the number of years we live in good health, free from disease and disability. This is the true goal, not just to add years to life, but to add life to years. The implications of this research are staggering and extend far beyond individual health. What would society look like if a healthy, active life of 100 years, 120 years became the norm? How would it change our concepts of career family? It's a question.